Now we're going to look at how the genetic code, our DNA, is used to code for proteins, which are the functional units in our body. Genes control these phenotypes through proteins, right? So we've looked at genotypes and phenotypes. The genes are your DNA, and the phenotypes are a result of proteins. So your phenotypes are the way you look, but these all result from the expression of genes in the form of proteins. This process of protein synthesis happens in two main steps. The first step is copying DNA into RNA, which is called transcription. This is taking the genetic code in the nucleus and copying it into a shorter piece of that genetic code called RNA that can leave the nucleus. Then, once we're in the cytoplasm, that RNA gets read to make proteins in a process called translation. The way that the messenger RNA is read into proteins is based on a triplet code called a codon. So codons are found on the molecule of messenger RNA, or mRNA. And a codon is made up of 3 bases, so it's called a triplet because it's made of 3 bases. So for every 3 nucleotides on the mRNA, that makes up 1 codon. For each codon, it is translated into a single amino acid. So in the picture, you can see the strand of DNA. This is the blue picture up here. And we use complementary base pairing to make a copy in the RNA. Remember, RNA doesn't have thymine, so anytime that A is present in the DNA, it's going to bind with a U in the RNA. C pairs with G, G pairs with C. So here's my messenger RNA, and every 3 nucleotides makes up a codon. Each triplet codon codes for 1 building block of the protein, an amino acid. There are 64 different codes that you can make with 3-letter triplets. So 61 of those code for an amino acid, one of which codes for the start, which is here. And then 3 of them code for stop codons. Those are these red markings over here. So this is called a codon chart. And we can use this to read our triplet code, and it shows us which amino acid is supposed to be added. So let's start with our start codon right here. So my codon says AUG. The way you read this is the first letter of the codon goes in this first circle, the second in the second circle, and then the third in the third circle. So we start with A. That's going to start here. I'll draw it. So A starts here, which goes to U, and then eventually G. That's going to lead me to methionine, which is what we have here, met. Go ahead and take a look at the second codon, AAG. So we start in the same spot in the beginning, A, only now the second letter is A, and the third letter is G. So that takes us to lysine, which is what we have here. And then the third codon codes for U, U, U. All right, so the first letter is U, the second letter is U, and the third letter is U. That leads us to phenylalanine, which is what we put in the third codon. The first step is transcription. This is when DNA gets copied into RNA. Remember that DNA is held in the nucleus, so transcription is happening in the nucleus. The DNA, so here's my strand of DNA, it has a region on it called the promoter. And this tells our body that there's a gene here that we want to copy. So the first thing that happens is we have an enzyme called RNA polymerase. RNA polymerase is going to bind to this promoter region because that's where the gene is going to begin. The next step, called elongation, occurs as the RNA polymerase moves along the DNA and adds RNA nucleotides. So here you can see the growing RNA molecule. And as this enzyme moves along the DNA, it's adding RNA one at a time, and it's using those complementary base pairing rules. Whenever it sees a T, it adds an A. When it sees a C, it adds a G. When it sees a G, it adds a C. And then when it sees an A, it adds a U, because RNA doesn't have thymine. And so this continues all along the length of the gene. Eventually, we get to the end of the gene. It has a terminator sequence, like this in red. The terminator sequence tells my polymerase that the gene is done. We've copied everything we need to copy to make the protein we want to make. So once the RNA polymerase reaches the end, it breaks off of the DNA and releases the mRNA strand into the nucleus. Because we have a nucleus, the mRNA actually has to undergo a couple of changes before it can exit to the cytoplasm. These 3 changes are called RNA processing, and they help to facilitate the RNA moving out of the nucleus and also to protect it once it leaves into the cytoplasm. The first thing that happens is we add a 5' cap. So we're adding a cap of guanines to the 5' end. And then we add a poly-A tail. So that adds a long string of adenines, A, on the 3' end. This protects the 2 ends from being digested when they get into the cytoplasm. The third step is called splicing, and that's happening here. These lightly shaded pink portions are called introns, which means intervening. Think about what the word intervening means. It means to get in the way or interrupt. So these 2 sequences don't actually code for what we want to make. So they get cut out. And then the exons, which are these dark pink parts, get spliced back together. They get glued back together. This makes a functional messenger RNA sequence that can now leave the cytoplasm. So the last step to making a protein is called translation. But before we start talking about translation, let's look at one of the main players. We have another type of RNA called transfer RNA. Transfer RNA is the molecule that actually does the translating. It reads the codons and then attaches the correct amino acids. It does this because it has an amino acid attached to it at the 3' end, that's this purple region. And it has a sequence down here called the anticodon. The anticodon can complementary base pair with the codon on the mRNA. So that's how the tRNA knows when it's supposed to add its amino acid. Remember that ribosomes are the organelles that make proteins, and ribosomes live in the cytoplasm. So it's important to get the genetic code out into the cytoplasm in the form of RNA. Ribosomes are also made up of RNA, called ribosomal RNA or rRNA. Let's look at the anatomy of our ribosome here. It has 2 sites. It has the P site here, which is going to be where the tRNA holds the growing protein. And then it has the A site here, which is where new tRNAs are going to arrive, A for arrive, as the protein gets built. So we begin translation with a step called initiation. Remember that initiation means to begin or start something. The first thing that happens is the small portion of the ribosome binds to the messenger RNA. And it does this near the start codon. The first tRNA is going to complementary base pair with the start codon. See how where the start codon has an A, the anticodon has a U. Where the start codon has a U, the anticodon has an A. And where the start codon has a G, the anticodon has a C. Once that first tRNA is in place, then the large ribosomal subunit is going to be added, and we have a fully formed ribosome complex ready to start adding nucleic acids. The second step is called elongation. This is going to grow the peptide chain 1 molecule at a time. So I'm going to show you the cycle for adding each amino acid. So we have our tRNA, which is in the P site, and the tRNA in the P site is holding on to this long polypeptide or protein chain. Then a new tRNA is going to arrive to the A site. It's going to base pair with the codon in the A site. Remember, A for arrive. The next step is going to be forming the bond between the previous protein chain and the new amino acid. So we're going to form a peptide bond, right? That's the bond that proteins make, between this circular amino acid that was already added and this square amino acid that's being added now. So once this protein bond is formed, now we have this tRNA in the A site holding on to the long protein chain. Only we want to move this one over to the P site. So in the third step, translocation, the entire ribosome is going to move down. So the ribosome moves to the right, which pushes this tRNA to the left. And now we end up with the tRNA in the P site holding the protein chain. And the only difference from when we started is that the protein chain is longer by the one amino acid we just added. And this is going to continue over and over and over again until the codon in the A site is a stop codon. All right, so let's review this flow of information. Remember that the central dogma of the flow of information goes from DNA to RNA to protein. The first step in this is called transcription. So here we have transcription, where my RNA polymerase is complementary base pairing with DNA to make a strand of RNA. We're going to make a couple of changes to this RNA called RNA processing, and then it's going to leave the nucleus. When it leaves the nucleus, it's going to do this process of translation. We're going to start with the first nucleic acid codon and the first tRNA binding to the first start codon. Then we use elongation to grow the polypeptide chain one amino acid at a time. And then eventually, the stop codon will reach the A site, and everything will be broken apart and released. One of the really cool things about these tRNAs and the ribosomes is that they can be reused. So after we hit the stop codon, once everything's released, they can just go right back to the beginning of the messenger RNA and start all over again to make more copies of this protein.